Now, let's study about the mass spectrometry. Same way, I will show you one brief mass spec intro movie made by Royal Society of Chemistry to give you a general idea. After the short intro movie, we are going to move into more details.
Mass spectrometry allows chemists to weigh atoms and molecules not directly on a balance, but by measuring how ions formed from them are accelerated and deflected by electric and sometimes magnetic fields. There are many types of mass spectrometer, but they all work on this basic principle. This instrument is called a magnetic sector instrument. The sample is introduced here, and the positive ions are formed here by bombardment of the sample with a beam of electrons. The ions are then accelerated by an electric field and then deflected in a curve by a further electric field. They then bend through a magnetic field and are further deflected by another electric field here. The ions then enter a detector here. By varying the electric and magnetic fields, ions of different mass can be made to enter the detector. As the ions are formed from molecules of the sample, they may fall apart or fragment. The ion of largest mass is often the parent or molecular ion, an ionized molecule that is not fragmented. Other peaks of smaller mass may represent charged fragments of this ion. The masses of these fragments may give clues as to the structure of the original molecule. The sample fits here, and ions pass out through this slit. The heater itself is here. The electrons are accelerating in this direction, and the ions stream in this direction through the slit. The positive ion beam passes between two charged curved plates, which deflect it in a curve so that it enters the area here, where an electromagnet produces a vertical magnetic field. This further deflects the beam in an arc of a circle. On emerging from the magnetic field, the beam is deflected again by another set of charged plates which direct it into the detector. The amount of deflection by the magnetic field depends on the mass of the ion, strictly its mass-to-charge ratio, but the vast majority of ions have only a single charge. The deflection also depends on the strength of the field. Heavier ions will not be deflected sufficiently in the magnetic field to reach the detector. Lighter ions will be deflected too much. During a run, the magnetic field is gradually increased so that ions of successively greater mass enter the detector.
How was the movie? Was it good? Okay, let's check some of those basic components of mass spec. The first part of mass spec is the ionization source part. And then it is connected to mass analyzer. And then final detector part. Historically, many different kinds of ionization methods have been developed. Still, modern proteomics approach, we need a very soft ionization. That's why currently most of those ionization methods we are utilizing are MALDI or ESI. And mass analyzer. The intro movie showed magnetic sector, but also we have a quadrupole, ion trap, or time of flight. Many more modern techniques are under development. Still, these are most common mass analyzers we are using at the moment. And by combining these two, usually we call those mass specs configuration. So let's say MALDI quadrupole, or ESI TOF, ESI ion trap, MALDI ion trap. Kind of any combinations we can make between ionization source and mass analyzer. This is a general scheme of mass spec application into protein analysis. In the beginning, we prepare cells or tissues and then extract out the protein. Usually we run the SS gel to purify those protein, and from the gel we cut the band and then digest those protein into peptides. These peptides can be injected into another chromatographic separation, and at the end it can be ionized and analyzed. In previous page, we said only one option of a mass analyzer, but mass spec can be connected together to make a tandem mass. Then it becomes more powerful. If you use only one mass spec, by injecting this peptide, we can see those precursor peptides. But we have a power in tandem mass. We can choose one peptide, and then further fragmentation we can induce, and then analyze that specific peptide to get more information in the second mass. By analyzing this fragmentation pattern, it is possible to determine the amino acid sequence of the specific peptide. That's why this tandem mass spec is so powerful for protein analysis. Tandem mass can have different configurations, and here are several very popular other configurations. One case is a triple quadruple. So one quadruple can act as a one mass spec, and then first one here and the last one here. Middle quadruple can act as Claisen chamber. So that means from first quadruple, one ion can be selected, and then it can be fragmented inside in this second quadruple and can be injected into the third one. So practically, mass analyzer is two, the first one and third one. This configuration what we call triple quadruple. Quadruple also can be connected to TOF as a second mass. In this configuration we call Q-TOF. Ion trap has a very interesting configuration. If you inject different peptides inside the ion trap, we can capture them all and then analyze those mass. Also, by controlling the electric field, we can choose only one peptide among those mixtures and break it up and analyze those sequences. So ion trap can be considered as not only mass, it can be multiple mass spec connected function it can display. For target ID and peptide sequencing, we have two different possibilities. One is utilizing tandem mass, Clasonally induced dissociation method, and then we can do peptide sequencing. The other possibility is a peptide mass fingerprinting method. In CID method, the peptide injected into fragmentation chamber, it can be fragmented by high energy. Now along those peptide sequences, there are different positions which can be fragmented. But interestingly, the main fragmentation happens in between this peptide bond. When the peptide bond is cleaved, two fragments can be generated. One is C-terminal and the other one is the N-terminal. So the N-terminal fragment we call B series and C-terminal fragments we call Y series. And different position it can form this terminal and this N-terminal, and also here. Also, other non-peptide part also can be fragmented, but fortunately to us, this G, X, or AC other fragment is a much lower chance than peptide bond fragmentation. That's why the analysis becomes much easier. So by analyzing this B series of a peptide or Y series of a peptide, it's possible to read out those amino acid sequence along line of this peptide. So from this mass spec, there is two chances. One is by reading those B sequence, or the other way by Y sequence, and then you can confirm the peptide sequence in two duplicate informations. This CID method can be applied to totally unknown peptide sequencing. That's why this is very powerful. Also can be called as a de novo sequencing. But the limitation is that requires a lot of analysis part of this mapping of those sequences one by one. In contrast, peptide mass fingerprinting can be much faster way utilizing genomic database. Now we are accumulating a lot of genomic informations by high-speed sequencing. And then if you have protein samples, let's just say proteomic sample, then we can take out those protein and then digest with enzymes like trypsin. And then we can get the mass spec data. In this case, if it is a single mass, still we can get different peptide mass. Now, from the genomic database, we can predict those product protein sequences. And also, utilizing those cleavage site information, we can predict those digested, fragmented, the peptide, the information. Now we can compare this in silico data with this experimental data and then match how many of those peptides are matching to each other. By utilizing these informations, we can identify the target protein. And this approach even can be applied to proteomic scale. And for the proteomic scale, two different approaches. One is the in-gel digestion, and the other one is MODPIT. MODPIT is multidimensional protein identification technology. Utilizing 2D gel separations, we can easily isolate out thousands of different proteins. And conventional method of protein analysis was take out the one protein and extract out that protein and digest and inject into the mass spec. So as that process takes a lot of efforts and time, so to automate the process, in-gel digestion method has been developed. By cutting out those bands and just put whole gel into the tube, and then destain and reduce to break up those disulfide bond, and digestion can be done inside the gel. That's why all the processes can be automated and can be much faster. Utilizing this in-gel digestion method, 96 samples handling is now practically possible. Now expanding that to the thousands of those spots are also possible. To make that speed even higher, now MODPIT technique has been developed. Rather than one by one of those protein separation in the beginning, mixtures of protein can be digested together. Now you can generate huge complex peptide mixtures here. To simplify the complexity, multi-dimensional column system has been applied. So first column can be strong cation exchange, for example. Second column can be reverse phase column. Now, only one portion of those fragments can be injected to the second column, and then full separation can be generated, and second portion injected again. This process mimics two-dimensional gel experiment, but not with the protein. That is done by peptide mixture levels. Using this ultra-fast proteomic analysis, now analyzing whole proteome one day is possible. Once you are successfully done with your target ID, maybe you want to know exact binding site of your drug in your protein. So here's one trick. Let's say this is your target protein. Now here, the target protein without your compound, this is with your compound. And there may be multiple cysteine around here, and one cysteine is directly binding to your drug. Here you can add alkylating reagent. This alkylating reagent can modify all the cysteines here, but in this case, the nearby this cysteine is directly binding to your molecule. That's why this molecule cannot alkylate it. By analyzing those peptide fragments, we can figure out which site of that protein is actually bound to small molecules. This technique can tell you exactly which cysteine is nearby to your drug compounds. That's how you can figure out the binding site of your drug in the given target proteins. Another interesting application of mass spec is affinity-based selection. So using this technique, you can do very high throughput screening of many compounds against one protein. Let's say you have many candidates small molecules which may bind to your target protein, and you mix them together and run the gel through size exclusion column, and unbound compound just left behind. Only protein-bound compounds can be isolated together with the protein. After you release this protein and then analyze those small molecules by mass spec, then you can get the strong binding small molecule very quickly. In principle, this approach, you can screen maybe hundreds of small molecules if they have a different mass, screening at one experiment and get out the most strong binding compound. It's quite an attractive method, isn't it? Now let's think about the proteomic approach for different samples. In 2D gel case, I showed you one example. You can label one protein with one color and the other protein with the other color, and then mix those samples together and then run one gel. That technique we called DIGE, D-I-G-E technique. Similar method we can do using mass spec. In mass spec, you can prepare one sample and the other sample, and you want to compare the relative protein amount. In this case, you can apply different mass tag. So for example, in this case, 8 deuterium compared to 8 proton, and maybe carbon-12 to carbon-13. And then if you mix two samples together, the same protein may have 9 mass difference. And then you can compare the relative amount of these proteins. So similar technique has been applied to different tag method, and currently most popular technique is so-called eye-track. And this eye-track are the method you can label 4 different mass for 4 different samples, and you can mix them all together. 4 samples can be analyzed together. Even further modified the eye-track, now we can analyze 8 samples together. That means that this technique can be applied to real proteomic scale. Whole cell extract, whole protein sets can be analyzed at the same time. If you combine together this proteomic analysis using mass spec with cellular thermal shift assays, let's compare. Previously, when you prepared these samples, and you needed to have one antibody to isolate out that specific protein and compare them. But now you can collect these samples from different temperature and then label them with a different mass tag. Now mix them together and analyze it. Then with a different temperature or isothermal analysis or different concentration or different time point, all different kind of experiment is possible. That means utilizing this thermal shift assay combined together mass spec, now proteomic approach for target ID or target characterization is possible. So the application of a mass spec in proteomic research, the potential is huge, and we can accelerate a lot of these difficult problems by combining this high throughput analysis method with other smart idea and can solve a lot of problems with much faster speed than before.